one cell, many cells. The continuity of cells, and thus individual organisms, and indeed whole species, relies upon accurately passing on genetic information from one cell generation to the next. This genetic information is contained within each organism and within every single cell, in structures inside the nucleus called chromosomes. The genetic information is coded in DNA molecules, which resemble a twisted zipper. In the process of DNA replication, this molecular zipper unzips to leave half the original molecule. The left half of the zipper uncoils to leave a chain of nucleotides, consisting of a phosphate, a sugar, and one of 4 possible purine or pyrimidine molecules. The spiral structure imposes strict limitations on the molecules, which can form the rungs of the DNA ladder. The bonding arrangement between purines and pyrimidines further limits which particular molecules can link together. This strict ordering, known as complementarity, means that the left hand of the zipper can exactly recreate the right hand half out of free-floating nucleotides in the nucleus, while the old right hand rail produces a new left-hand one. This produces 2 DNA molecules, one for each cell during the process of cell division. DNA clearly contains the coding that allows its own reproduction. If organisms were built entirely of DNA, life would need no further decoding. But the DNA structure also contains the genetic coding for the construction of protein. How does this more complex decoding and manufacturing process take place? The first step is the synthesis of ribonucleic acid, or RNA. While both RNA and DNA are long-chain molecules made up of linear sequences of nucleotides, ribonucleic acid differs from deoxyribonucleic acid in a number of ways. The difference in name comes from the sugar molecule along the rails. DNA is built with deoxyribose, whereas RNA is built with ribose, which has one more atom of oxygen. DNA is a double helix, while RNA is a single helix. Unwound, RNA is like a ladder cut in half lengthwise. RNA is smaller than DNA, but it's much more plentiful. DNA, at least in eukaryotic cells, is confined to the nucleus, while RNA enjoys the freedom of the cell, so to speak, and is found in both the nucleus and the cytoplasm around it. Lastly, there is one type of DNA, but 3 distinct types of ribonucleic acid. These are messenger RNA, transfer RNA, and ribosomal RNA, each identified in accordance with their function, and each playing a specific and irreplaceable role in the synthesis of the cell's proteins. As in the case of DNA replication, RNA synthesis begins with the unzipping and unwinding of the DNA double helix. But adenine does not bond with a free-floating thymine nucleotide. Uracil takes the place of thymine in bonding with adenine. RNA synthesis does not occur along the whole DNA strand, but only in limited sections. After the synthesis is complete, the resulting messenger RNA strand is cast free of the DNA template. From the DNA blueprints in the cell, messenger RNA carries genetic information out of the nucleus to the sites of protein synthesis, called ribosomes, scattered throughout the cell. In DNA replication, all that is required is to copy nucleotide for nucleotide, the complementary sequences called for by the left hand or right hand strand of the unzipped DNA helix. But for the purposes of protein synthesis, the information in the genetic code of the cell must be read in a way that relates to the amino acid units that will eventually make up any given protein. Once the genetic code has been transcribed to RNA, it seems that the code is read from the messenger RNA 3 nucleotides at a time. Each triplet of nucleotides is called a codon, and each codon codes for one of the 20 amino acids. Thus, the sequence GCU codes for the amino acid alanine. GGA codes for glycine. CAC codes for histidine, and so on. Reading the genetic code in triplets appears to be the most efficient means of relating the linear nucleotide sequence in messenger RNA to the amino acids used in protein synthesis. Pairs of nucleotides would provide 16 possible combinations, too few to account for the necessary 20 amino acids. And quadruplets would provide far more combinations than are needed. Nucleotide triplets, however, provide 64 possible combinations, a comfortable margin for the 20 amino acids that must be coded for. Indeed, some amino acids can be coded for by as many as 6 different nucleotide triplets. These 6, for example, all code for one amino acid, leucine. Of the 64 combinations, 3 codons do not code for any amino acid. These codons serve as terminators. During the transcription of the DNA nucleotide sequence, a codon such as UAG signals the end of the RNA transcription process. Another codon, AUG, is unique in the genetic code. It serves a dual purpose. It codes for the amino acid methionine. At the same time, it is found at the beginning of all RNA messenger strands, evidently providing a start signal for the messenger RNA transcription process. How the two purposes are distinguished from one another in the process of RNA synthesis is not yet known. There's an intriguing postscript to the transcription of messenger RNA. Almost immediately following the completion of the transcription, there attaches to the messenger RNA a tail consisting of some 200 adenine nucleotides. Messenger RNAs without the tail last only a few minutes in the cytoplasm before being broken down by enzymes, while those with a tail can be stable for hours or even days. Once the messenger RNA has passed through the nuclear membrane, it links up with the extremely small ribosome. It is here that the genetic instructions carried by the messenger RNA are received, and with the vital assistance of transfer RNA, are translated into functional proteins.